U.S. Forward.
Yeah. I've got multiple projects here going on.
Okay, good afternoon. Welcome to the May 2025 meeting of the Houston County Historical Society. We will officially call the meeting to order. If you will, please silence your phones or put them on vibrate or whatever you need to do to still be able to access them. There is a paper going around. If you haven't signed in, please do so. We will start off by the pledge to the flag, led by Miss Linda Smith.
Pledge allegiance to the flag of the United States of America and to the republic for which it stands, one nation, under God, indivisible, with liberty and justice for all.
Thank you, Madam Secretary. Meeting minutes. Everybody get their minutes okay? Have any questions, problems, corrections? Ms. Betsy, did you see any problems with the minutes?
No.
Everything you said was quoted correctly. Good job,
I'll
Melissa.
just say this, and I'm the one who does the minutes, and I had to go back and I watched the recording of each meeting.
Yeah.
And I hate to say it, but a lot of the times I'm dreading it because it's like, oh, okay, I gotta get all this information. I was so looking forward to going back and watching your presentation.
Well, I got more of a kick out of it by reading it than, seeing it, so, good job. Are there any corrections to be made to the minutes? Corrections, additions, anything? Well, we will deem the minutes approved. Treasurer's report, Ms. Leah?
We had income of 74 cents in interest and expenses of $474, which leaves us a balance of $992.10 in our checking account. On the positive side, our CD got us $117 in interest, so we have a balance in the CD of $11,276.98.
Is there a motion to approve?
So moved.
Second?
Second.
All in favor? All right, any opposed? All right. Leah, you're on the list again about the flag update.
We have several, as some of you may know, Glenn he's not here because he's across the street, but the American Legion and the VFW put up flags at a number of the cemeteries in Houston County, and we have some that we need help with. So if there are any near where you live that you might like to help with, let me know.
Because we have 236 cemeteries in Houston County.
We don't have,
But not every cemetery has a veteran buried there.
we have just over 1,200 veterans buried here in Houston County that we know of.
And some just have one veteran, some have none, some have...
Right, a lot have one, and there's one big one that's got, the biggest is Cedar Hill has got over 200, about 200 veterans.
And then Oak.
And the DAR takes care of that for us. Rick Conley takes care of that. Oak Heights, that, sorry Linda, wherever you are, sorry. He has the list of the flags.
Oh, Mitchell, yes.
Mitchell has the list for Oak Hill, and so if anybody's willing to help out, I'm sure.
And that's the second largest.
That's the second largest, yes.
So if we get those two, we've got a good majority of them.
That's
And the
almost
others are
400 of
scattered.
them, yeah.
So we enlist your help with some of the others. Okay, Jackie and Donald, do you have any updates on the Korean and Vietnam Veterans Project?
We're still collecting information on all our Korean and Vietnam veterans, and if you know anyone who's a vet, please get in contact with us. We'd love to have pictures and information about their time in service
Maybe a program in the future?
in the future.
Maybe part one, and there's so many we may have to do a part two.
And I didn't have a chance to speak with her, but Kathy Freed, she used to be the VSO in Stewart County. She's very active with all three of the organizations. She is looking for the living Korean veterans.
Right.
Have you
I was in
spoken with
touch
her?
Yeah.
Okay.
Which there aren't that many.
No, there's none.
Next on the list, Miss Melissa, do you have an update from the archives and museum?
Updates since last month. Again, Archives is going strong. We get donations all the time. And I mean, when I talk about donations, people will just walk in and they go, do you want this? And of course, anybody that knows me knows I don't hardly ever say no unless I can't fit it in the room. But today, I want to share a couple of donations with you. But this one came in today. This one is a mailbag, and Clint Reynolds donated it today, so I'm going to ask him if he will tell us about this mailbag.
I found it in the Rokas house, and I'm assuming that it belonged to Leon McCauley, because he was a mail carrier for many years at Stewart, and evidently it was left in the house when they sold it to the Rokases.
And you said it said what?
It's the U.S. mails and
Yeah,
stamped in the
it says,
leather.
you can't see it, it's embossed, this is leather. And so,
I think it's only half of it.
yeah, I was telling Clint that if and when we move to the Armory, the Archives and Museum to the Armory, we're gonna transfer the Stewart Post Office that is right now on display at the fairgrounds to that new location. And we have lots of memorabilia and artifacts and things like that to display with that post office, and so this is gonna be a great one to put with it. And so people are also asking, what kind of stuff do you take? We take anything and everything. Leah can tell you, when they walk in, I say, just bring it. So this came in today. Thank you, Mr. Plant, for doing that for us. Appreciate it. The other item that Leah has been hearing me talk about over and over was donated. Go over here. It was donated a few weeks ago by Miss Wilma Wyatt Nichols. She, if any of y'all know Miss Wilma, okay. She was actually a caretaker for Miss Lorraine Boone. And so Miss Lorraine Boone gave her several things before she died. So she brought in some of those things. We have a lot of records, photographs, memorabilia, artifacts from the Boone family. We had one of their descendants donate a lot of stuff, but we still have stuff that trickle in from the Boone family. They're a very well-known, a very well connected family in the county. But this particular item, and every time I talk about it, Leah's probably thinking she says the same thing. It's absolutely beautiful. And not everything that comes into the archives is beautiful. But this is absolutely beautiful. This is an item that Mr. Eric Rosher gave to Miss Lorraine Boone as a Christmas present in 1928. He bought it at a jewelry store in Nashville that no longer survives. And it is in almost perfect condition. And so I wanted to bring it and share it with you. It is from the B.H. Stife Jewelry Company in Nashville. Oh. It is a purse. It's a beaded purse. It has a little bit of the fringe missing here, but other than that, I have looked it over and I honestly don't even see one bead missing. Looks the same on this side. And it does have some staining up here from age and being exposed to moisture. But I just think it's absolutely beautiful. It is. And when you open it up, it is perfect inside. I don't think she ever even carried it. I can't imagine carrying this, but all the stuff I stuff in my purse. But
What does it represent?
to me, I don't know. There's two ladies here, and they look like they're wearing colonial dress. In fact, they look almost like colors of the American flag. I don't know. I don't even know. The building, believe it or not, reminds me of the Kremlin from Russia when you look at it. But I have no idea. There's no tag in there saying who made it, other than it came from this particular jewelry company in Nashville. So I will leave it on display. And you are welcome to look at it. And you're welcome to pick it up by the handle there, because like I said, it's pretty good condition to be as old as it is. I actually ran into Miss Ann Hackler today at the Piggly Wiggly, and I told her she needed to come by and see this, and her sister Sue was with her. And I told her, I said, your grandfather Eric Rosher gave this to Lorraine Boone for Christmas. So she says, oh, do you think there's something there? I said, I don't know. I think they're looking for any kind of scandal. They love the scandals. So, but yeah, so please look at that. And I've also got the mail pouch here. I'll put it down in front. But, you know, I talk to you all the time about not throwing anything away, donating it. Please do that. Whatever you find, even if you don't think I want it, you don't know if I want it, bring it to us, because people do that and they go, well, I was going to throw that away, and it's something we really need to keep and hang on to for our local history. So just keep us in mind. Thank you. It is heavy.
Right here. Okay, is there anything else anybody needs to discuss? If not, we will move on with our speaker, Mr. Steve Black. Mr. Black and his wife Karen comes to Houston County from California. They live in the western part of Houston County, in the White Oak Creek area. They've been there about 10 years, and he has graciously agreed to do our program today. Mr. Black.
Good evening, and thank y'all for coming. Yeah, we moved here about 10 years ago. My background is, I'm an engineer, an electrical engineer. I know nothing about trees or botany other than what I've learned since then. My wife's also an electrical engineer. She worked on the space shuttle. I worked in electro-optics and night vision, so I might have built something that went up in space, but I actually did. There's something that's on its way to one of the planets around Jupiter that I worked on. So you'll get there in another year or two. It's a long drive. We live out on Priestley Irwin Lane, not too far from White Oak Creek. And Karen, myself, 2 dogs, and some chickens, and a cat that's don't know where she went today. Tonight's talk is going to be about the American Chestnut. We're both members of the American Chestnut Foundation, been so for many years. I guess it's kind of a inherited thing. I know her dad, who grew up down in the Hickman County area, talked about chestnuts. He was the youngest of 10 children. His grandfather was a Civil War veteran, captured in Fort Donelson, so a few local ties here. Anyway, we've been involved in chestnuts, and now that we're here, our farm, we've got a large chestnut tree on the farm. It's not American, but it produces very delicious nuts, and we are attempting to raise American chestnuts. So, talk about chestnuts. The chestnut story is certainly a sad story. The mission of the American Chestnut Foundation is to restore the chestnut back into our woodlands. And to do that, we're having to develop a disease-resistant chestnut. This illustrates the range of the chestnut where it used to live, as best we can tell. It starts down, and this is what we call kind of the southern population. It includes us right around in here. The central population, which includes the Appalachians, and then up into the New England area. And we think of the populations different. The trees all look the same, but there's subtle genetic differences. And when we work restoration, we have to maintain those differences, because you don't want a tree, for instance, if we took southern genetics and moved them up in New England, the tree would flower too early, and that would not be successful. So anyway, that's the native range. In some areas, it was 1 of 4 trees was a chestnut. It was a massive tree. They could get to be 100 feet tall, 15-foot diameter, sometimes called the redwood of the east. Very long-lived,
Pretty straight.
very straight. Their growth characteristics in some ways are similar to poplar, and they can hold their own in growth rate with poplar. In a good site, they grow like mad. I've got some seeds that just sprouted last month, and they're already like this tall. And they were a very useful tree. Obviously for wildlife, the turkeys, deer, passenger pigeons, squirrels. If you're trying to raise them, you know about squirrels. And humans. It was a major cash crop for the people that lived in Appalachia. They'd gather the chestnuts in the fall, send them to the cities. It might be their only cash crop. They'd let their pigs loose in the woods and they would fatten them on them. The timber, you'll still find chestnut barns, chestnut buildings. The timber is high in tannin and is fairly rot resistant. And they did use the bark for canning hides. So what happened? Somewhere I got a cursor. So over the range, in the late 1800s, some trees that were noticed that were dying. But around 1904, around the Bronx, they started seeing dead chestnut trees. And it sped fairly rapidly, entered Tennessee in the late 30s, and by the 1950s, it covered the entire range. Moved very fast. There were attempts to stop the blight by cutting trees, cutting every chestnut tree in an area. What they didn't realize is it was also infecting oaks. It would affect scarlet oaks and would live in the forest, the litter on the forest floor. So it still had hosts. It can be carried by the wind, the spores. So it was quite the tragedy. And left behind were the skeletons, approximately 4 billion. And the suspect, actually it's not a suspect, it's pretty well convicted, was blight, we call it Cryphonecter pharisaica. It came in chestnut trees. People had brought Japanese and Chinese chestnut trees from Asia, and one of those trees was infected with the blight and brought it to this country. It's a foreign pathogen, and the native chestnut here had not evolved with it, did not have any defense mechanism. What it does is it causes these cankers to form on the trunk, and they eventually go all the way around the trunk, and they interrupt the flow of water and nutrients between the root and the top of the tree and kill the tree. Another interesting thing is what you're seeing here is these are actually relatively young trees that are sprouting from the stump of a dead chestnut. And so while the chestnut tree is gone, there are still millions of chestnuts. They're just very small. They sprout from the stump. They get to 1 inch to 2 inch in diameter generally. Then the blight gets them, they die back, and then the stump sends up a new sprout. And this has been going on for a century now. So current kind of estimate of number of chestnut trees, it's a rough estimate of where they are. There are a few remnant trees. There's some trees on the West Coast in Oregon and Washington where settlers brought nuts with them and planted them, and they're outside of the range of the blight and hasn't been able so far to cross the desert. There was a remnant population, it's hard to see, it's over here, of, again, a settler that carried some nuts and went up into the Midwest and planted them. And there was a grove there that unfortunately the blight got to it about 20 years ago and it's largely destroyed. The other challenge with chestnut is a few of these sprouts get big enough to flower, but the tree is not self-fertile. It requires another tree to pollinate it. So suddenly you get the odds of, it's unlikely to have one of them flower, it's even more unlikely to have two of them. So they're not able to reproduce. So we use the term functionally extinct. There's still lots of them there, but they're not able to reproduce themselves. And unfortunately, I mean, these root systems are now a century or more old, and we're starting to lose some of the root systems, which is our genetic diversity. So there's more than one kind of chestnut. There's American chestnut. It's tall, not resistant to blight. It's be a very large tree. It can be dominant in our forests. Then there's the Asian chestnuts. There's the, we call, when you see Castaña, that's the family of chestnut. So they all carry that Castaña in front. Dentata is the American chestnut, the botanical name. Melisma is Chinese, and Crenata is Japanese. The Asian chestnuts are very resistant to blight because they grew up with it. But the tree is more of an orchard tree. So it's a spreading, doesn't grow into a tall tree. And so that's been its own problem. In talking about the species, here's American chestnut, the nuts, the Chinese, European chestnut, and the Japanese chestnut, and the corresponding leaves. And we'll go into a little more detail on leaves. So if you think you found an American chestnut, first thing to look at is the leaf. You'll see the leaf will tend to have a dull, matte appearance. It's not shiny. It's fairly thin. The teeth, or we call them dentations, almost look like a wave cresting. The leaf is narrow and kind of comes into the stem up here at kind of a gradual angle. So that's always the first thing when somebody says, I found a chestnut, is I can look at the leaves, because you can learn a lot by looking at the leaves. The twig tends to be red to chestnut brown of all things. The buds are smooth and pointed. Now the leaf can take, like any tree leaf, there's a lot of variability in leaves. Whether they're in the sun or shade leaves and sun leaves, shade leaves tend to be bigger. How old they are, how old the tree is. It's going to give you an idea of some of the variety of American chestnut leaves. Now the other one that you're likely, in fact, more likely to run into is a Chinese chestnut, or a mixture of it. TVA had a program in the 50s where they had a nursery and they were raising them and distributing them as a replacement for American chestnut. And so there's quite a few in the area. We have one on our farm. First thing you notice, the leaf is shiny. If you feel of it, it's leathery, it's thicker. There's fine hairs on the back, and the little dentations or spikes on the side don't hook over. The branches tend to have a little bit of fuzz on them. So when you look at them, they're different. So finding them, later this month is a great time to chestnut hunt. The trees are in bloom. They'll just be covered in the white, creamy white catkins. And they give kind of a musty smell. I mean, you know, if you smell something that's a little odd that time of year, it's probably a chestnut nearby. So they're easy to find. If they're around, I drive around that time of year just seeing if I can spot one. The other thing you can look for is the burrs. Burrs are 2 to 3 inches across, very spiky. My dogs will not go under our chestnut tree when there's burrs on the ground. They're very uncomfortable. You'll find remnants of the burrs all the time. It's relatively uncommon to find nuts if you've got squirrels around, because the squirrels and deer and most everything like the nuts. Chestnut, I was asked the other day about chestnut wood. It's often can be confused with red oak. And a key thing is if you look at the end grain, red oak has what you call medullary rays. They're lines that run across the growth rings. They're there, they allow the tree to transport moisture in and out. It's also why you don't use red oak for barrels that hold liquid, because they leak. Chestnut does not have the medullary rays. The growth rings tend to be fairly wide apart. The wood, it's kind of similar looking. That's a very old piece of chestnut. The wood can look similar. The presence of the rays is really the giveaway there. So what have we been doing the last 100, it's over 100 years now since this disaster happened. In the 1900s, the first plan was we'll replace them with Chinese and move on. But the Chinese tree, if you recall, grows like kind of like an orchard tree, got branches going out sideways. It can't compete with a poplar or an oak. And so you get them in the woods, they don't grow well. The other trees overshadow them and they're in the shade. Well, that didn't work. So then the research station, I think Connecticut Research Station, had a breeding program, and they took every flavor of chestnut, whether it's European, Chinese, even chinkapins. Chinkapin is a member of the chestnut family. It's got a single small nut. And they tried all different mixtures of them, breeding them together, hoping to find something that sort of worked. It didn't. In the 50s, we're in the atomic age, and somebody had the idea, we'll take a bunch of chestnuts and radiate them and see if we can shock them into doing something right. That didn't work. In the 70s, there was a lot of work with what's called hypovirulence, and that was developing a virus that basically made the blight sick, so it wasn't as vicious. It didn't attack the chestnut as much. And we were able to make that work. The problem is, for reasons we don't understand, it doesn't want to propagate in North America. You can go and treat a tree with it, and that tree will survive, but it won't propagate to the tree next to it. So in terms of something you could let loose in the woods and it would solve the problem, it doesn't solve the problem. Surprisingly, it works in Europe. So you will see European chestnuts that are surviving, and that's because of hypervirulence. So there's something about the biological environment in Europe versus America that is different. We thought about trying to engineer a virus, but especially after COVID, I think people would not be very receptive to engineering a virus and letting it loose. In the 1980s, we went to what's called backcross breeding, and this was based on an assumption that there's a couple of genes in Chinese chestnut that control resistance. So we'll cross American with Chinese. We don't quite like what we get, but we'll take the most resistant of those crosses, cross them with American, take the most resistant of that product, cross it again, and we'll get something that's 99% American and is disease resistant. Sounded good. What we found was each successive cross, we lost disease resistance. What we didn't know, and we had no way of knowing, is that there's more than 3 genes that control disease resistant in Chinese and the Asian chestnuts. It wasn't until the 90s that they developed the ability to sequence the DNA of an organic material and we started to be able to learn more about it. So, and that's where the American Chestnut Foundation started, was with the back crossbreeding programs. We've been at it 43 years now. So in the 1990s, and I'm not going to go into a lot of detail here, that would be a whole nother talk, is transgenic chestnuts, is we did some work with transgenics. It took a decade just to learn how to grow a tree from a single cell. So there was a lot of foundational work there. We had some limited success, but my takeaway is we learned how much we didn't know. And you have to have really an atomic level understanding of the disease. So we'll go into that more in a later talk if there's interest. So current efforts, and I'm going to just focus on this side, is what we call genomically aided breeding, or recurrent genomic selection. And it leverages our ability to get the DNA of a chestnut tree. We've got a large database. We've got over 3,000 trees that we have gone and got their genetics. I mean, that's enough to populate a little bit of ancestry if you're into that. And then use the genetic data to drive our breeding program. And we've also identified some surviving American chestnuts that have some level of resistance. And we've learned how to find those genetically too. And then repeat this, so we'll talk about that. So, people have been breeding plants, animals forever to try to make them more suitable for whatever people have in mind. Recurrent genomic selection uses the genes of what you're breeding to help drive the selection. And that's important here. In many cases, you can just use what are called phenotypes. Phenotypes, like one of my phenotypes, I have blue eyes, is a characteristic. So like if you're breeding chickens to be a certain, to grow fast, you can weigh the chicken. And pretty quickly you can develop the phenotype versus the genetics behind it and drive your breeding program. With disease resistance, how fast a tree gets sick is hard to measure. We've got ways of doing that, but we're using the genetics of individual trees to help figure out which ones we want to breed. So this just gives you an idea of the evolution of chicken from what it was in 1957 to 2005. And that's why chicken has not exploded in price as much as other things, is because we've gotten better at raising big chickens. Another one was, in fact, we're teamed with the University of South Carolina, who's done a lot of this work with pine tree, improved genetics, blah pine. And this is a test plot. These are non-improved controls, and this is one of the planting of improved trees. And they're taller, they grow faster, but very similar process to what we're using. So we believe you can get there from here. So the first step of the recurrent selection is choosing the parents. And so for virtually every tree in our database, we've gone and looked at what we call phenotypical data. How did it behave? Is it alive? That's the first step. How big is the canker? I mean, is it covered the whole trunk, or is it contained? Did the canker sink? Is it in like it's a, I don't know how to say it, but is the tree trying to fight it? Is the bark of the tree just falling apart, exposed wood? Is there spores of the blight? If there's spores, that means the blight is thriving and is able to give off, is reproducing. So that all and other factors go into a bunch of computations to yield one number, which we call the blight resistant index. 0 is not resistant. 100 was where Chinese was. And we kind of built this ruler fairly early on, and then we keep adding trees to it and adding trees to it. And we had some that ended up worse than 0, so, and we had some that were better, but we keep the same yardstick. Here's where American chestnut is, and that's why we're working on the problem. It's got basically no resistance. But the good news is we've got a population up here of trees that have got elevated level of resistance and have got American ancestry that's like better than 70%. So that's kind of what we're using for our breeding population right now. So that helps us identify parents. Then we go forth and we find the trees. And we collect the pollen and freeze it. And this can be a 2-year process because a tree that, say we want to cross a tree in Pennsylvania with a tree in North Carolina, they may not flower at the right times. So we'll collect the pollen in one year, freeze it, and the next year go and pollinate the other tree when it's ready. So we've got a large data bank of pollen that we've collected, and each thing is labeled as to where it came from and the genetics of the tree. And then we go forth, and before a tree starts to bloom, we'll go out with a bucket truck or ladders and bag the flowers. Not all of them, but a sizable number of them. Because we don't want them to be pollinated by other pollen. So we'll go ahead and literally put a paper bag over them. And then we come back when the tree is, when it's flowering, and we'll hand pollinate those female flowers with the pollen we've gathered and rebag it, and then come back in the fall and harvest the nuts and try to beat the squirrels. So one of the things that came out of our collaboration with the University of South Carolina was some of the software they've used to look at doing this kind of genomically driven breeding, and we applied it to our data set. And this is an estimate of kind of our first year of doing it, which was in 2023. This is blight resistance of kind of where this is just breeding, letting trees breed, do whatever they're going to do. And they're kind of all over the map. There's some that are very resistant, but most of them aren't. This is where we think the population is going to be after one cycle. So we've tightened it up and moved the center point of the distribution from there up to there. Now, something I hadn't mentioned is we got 2 problems. We got another problem that came from Madagascar in the 1790s from the cinnamon groves. It's called Phytophthora cinnamomi. It is a soil-borne homicide or water mold that is an absolute plague worldwide. I mean, some of the big fires a couple of years ago in Australia is because this has gotten loose into their wildlands and it's killed most everything. Where we used to live in California, we raised avocados. We don't do that anymore. It killed all the avocado trees. So it is present, it kills chestnuts, it also kills oak and other things. So we're trying to optimize for some resistance to that. We don't have as much data on Phytophthora resistance or PRR, because we don't like to say Phytophthora cinnamomi 100 times. PRR, we don't have as much data, so we're trying to optimize for it. The major focus is blight resistance. If we just optimize for Phytophthora, we do a little better on it, but we hurt ourselves a lot on blight. We don't know whether that's real or not, or just the data is insufficient. But we think in the first generation, we're going to roughly double the resistance. So what do we do with these trees? We raise a lot of trees. So 2023, we pollinated, got nuts at the end of 2023. In 2024, we planted the nuts and turned them into seedlings. So we take about a quarter of those and they go into our seed orchards. We run a number of seed orchards. That's where we get the seeds that we use for future development. We give them to our members. We have seedling distribution events and so on. The seed orchards, we got 3 of them. One of them is in Meadowview, Virginia. One of them is in North Carolina. And then there's the Ruth Cochran Orchard in Tennessee. That's backed by Chattanooga. We have a collaboration with the University of Tennessee, Chattanooga, and Dr. Bill Craddock there. And then some go into field progeny tests. The field tests are very important because we can grow these in in nurseries, and they're in very sheltered environment. But what really matters in the end is, can they survive in the forest? So we got to get them out into a wooded environment where we can see, can they compete, can they survive? And so we're constantly doing test planning. We do some in Meadowview and around the orchards, and we got some that we haven't quite figured out what to do with them. We were planning on doing some work in North Carolina, but got pretty well derailed by the hurricane. That just the area we were going to plant was just a mess. So you notice this last that group was 1,400 trees. So next year we got more nuts. Our trees are getting bigger. And so we have about 6,700. Because it takes a lot of trials to develop, and then trials and winnowing down to get the improvements we're looking for. So again, more are going into seed orchards, about 800. So there's another thing we call a small stem assay. So the way we used to test for blight is we'd let a tree grow until it's 8 to 10 years old, and then we put a wound in its trunk, insert the blight, and then see what happened. And the problem is you have to wait 8 to 10 years. So some folks developed a technique where we just cut either the tree off or cut a branch off and dip the cut end in the branch, and we look at how fast the blight moves down the stem. And we've been able to correlate that with resistance. And it's an easier thing to measure, and you can do it on a 1-year-old seedling. And a lot of the trees I plant on our farm, you look at them and they've had their top cut off. That's because they went through this assay. But the trees grow up. I mean, I'm not growing them for timber or anything. They grow fine. But the small stem assay is key for telling us whether our model is accurate, because it gives us a blight resistance number that we can feed back in and see whether we get the answer we expect. Field trials. Again, we're now looking at getting about 3,200 trees out. We're still looking for sites for that. I've been talking to Caleb up at the Stewart State Forest, and we think we're going to try to do one in there. He's also found what he thinks is at least one chestnut tree in the forest, so trying to get more going in that direction. And then photophic root rot, or PRR. That's a problem to even test for, because we don't want to bring that pathogen into any of our orchards. Because once it's there, you can't get rid of it. We used to do a lot of this work with the Department of Agriculture, but some of the changes in the last couple of months there, the facility where we did that testing is currently not open, so we're trying to figure out something different there. But it's got to be done off-site in a site that's contaminated with the PR organism already, so you don't want to spread it. And we're very much a citizen science organization. I think the staff is maybe 20 people in the national organization that help coordinate the science, and we've got run the Meadowview research facility in those orchards. But all the rest, it's folks like Karen, myself, and anybody here in this room that's interested in jumping in. But we're very chapter driven. We've got plantings. This will have some more dots here when they update it, which is the plantings on our place. But You know, all these are areas that kind of cover the native range and where we've got people that are interested in making it happen. You know, the typical cycle is we'll find a location and then do some trial plantings just to see how it goes. And then raise some trees for that site, plant them, and then watch them for at least 10 years and just see how they grow. And I mean, there's trees now we've been watching for 30 and 40 years, so you gotta be patient. The only thing trees do fast, I've learned, is die. They got their own timetable. But one of the purposes, again, for the field trials is, are they competitive in the forest? Because if they're not competitive, it doesn't matter how resistant they are, they will not be successful. These are some of our trees. I think this is actually at the planting at Penn State. It's an earlier planting. We've got some nice sized trees going there, good form. We do have some very good looking trees. Our big focus now is to get the yield of good looking trees up. It isn't going to go anywhere if it's a 1 out of 100 sort of thing. So we got to do better now. And it's all about chestnuts, but as I watch things, it's not just chestnut that's having to deal with this. It was the first, but since then, the American elm that was kind of lined the streets in many of our these eastern cities is largely gone. Dutch elm disease. The butternut is largely gone, or at least severely impacted by butternut canker. We're watching the ash trees since early 2000 when the emerald ash borer appeared. It's now in Williamson County and Dixon County, I believe, so it's coming our way. That's been just a plague for ash trees. It's wiped out millions of acres of ash. Beech, this was just discovered about 5 years ago, beech leaf disease. It's caused by a small nematode, a microscopic worm. It probably gets carried by birds and so on, and is starting to kill beech trees. Eastern hemlock that line the Appalachians, they're in very bad shape due to the woolly adelgid. It's a small, almost aphid-like insect that sucks the sap and just weakens the trees till they die. Oak, there's gypsy moth, which is an imported moth, and then another fungal disease called oak wilt. Walnut is dealing with thousand canker disease. It hasn't gotten here yet, but it is hard on walnuts. Sycamore, we just cut our last large sycamore down. It was dying. It's what's called hypoxylon canker, I believe. It's another invasive disease. Oak and chestnut are both suffering from Phytophthora cinnamomea, as many others are. And then you may notice that now in the spring we don't see as many dogwood. That's dogwood anthracnose, which is another, I think that's more of a virus, but, or it's a bacterial infection that's largely eliminated dogwood in many areas. And unfortunately, most of these threats involve invasive things that are here because of us. We bring in plants from other parts of the world and they hitchhike. We visit areas and they come in on our shoes. We're moving around, we're mobile, that's us. But it has some negative effects. And I'm concerned what our forests are gonna look like in a century. Because there's a lot of this going on and there's more that I haven't captured here. So what can you do to help? You can join the American Chestnut Foundation, learn a lot about chestnuts. There's a link to our website. That's the Tennessee chapter there. Plant something, whether it's pure American or hybrids. help someone in their orchard. I know, gosh, you didn't know this, but hurt my back last year and boy, I needed help. Got behind on the weeding and other problems. And find trees. And our genetic database for American chestnut for the southeastern population, that's us, is rather thin. We need to find more chestnut trees, and the process is we'll go look at it, kind of looking at the cues that we went through in terms of leaf and so on. If it looks like it's an American, then we'll get a leaf sample, go get its DNA checked, and that'll tell us for sure what it is. And then we come back when it's dormant, get a leaf, get a twig, then we'll go graft it and capture that tree's genetics in our orchard. They keep looking for them. I run around the county this time of year when they start flowering, people tell me, hey, I found them. Unfortunately, so far, all I've found is Chinese or Chinese hybrids. I know there's some here, it's just finding them. And why plant Native Americans? It's going to be an exercise in frustration because they will die. But if it survives a couple years where it's well established, it will resprout. And by doing this, you've collected the genetics of an American tree and preserved it potentially indefinitely, and helping to backfill for those century-old root systems that we're starting to lose. When they start producing nuts, and they do it at a relatively early age, I've had trees start producing at like 4 or 5 years. I mean, it's not a big crop, but it's something. And you learn about how to care for them, and sadly, we'll learn how fast they can die. You've been so quiet. Questions?
I got a question.
Go for it.
I understand which side you're coming from, you know, do you know of any testing or researching that's going into the blight itself? On another level, if it's just, you can't cure it, I guess.
So that gets us into the transgenic side, and how a pathogen interacts, say, with a tree. It turns out it's amazingly complex. You have what are called genes, makes your eyes blue or whatever. There's also what's called promoters. And those are the control mechanisms that control that gene and tell whether it's active in doing something or not. For instance, you plant a plum tree. In that tree's genetics is a promoter that tells it to wait until I've had, say, 800 hours of cold before I put my leaves out. There's another promoter that somehow says, I'm 5 or 6 years old. You can start having fruit. And we literally have been able to turn off those promoters and have a seedling that can barely stand up, flower, and try to produce fruit. So, but where I'm going with that is the pathogen can reach in and trigger or defeat those promoters. And the tree reaches in and tries to mess with the pathogen's genetics. One of Steve's theories, and Steve is not a geneticist by any means, is that I think the chestnut is misidentifying this pathogen as something else. Because trees have different defense mechanisms, and they look at, oh, I recognize that, that's a flathead borer. I'm going to flood the burrow with resin and kill it. That's a mistletoe type parasite. I'm going to surround its roots with dead tissue and it won't be able to feed on me. If it uses that latter defense mechanism on a necrotropic fungus, like chestnut blight, that feeds on dead tissue, all the tree has done is, by that response, has fed the parasite, has fed the blight. And so effectively, the tree has committed suicide. And the faster the tree tries to defeat the blight, the faster it grows. So, getting that level of understanding, the early transgenic work we did was based on an observation that the tree gives off, or the blight gives off, oxalic acid, which is, I think, is used in like tanning leather and other things, but it's toxic to the tree. And so they knew about a gene in wheat that detoxifies oxalic acid. And they knew that because you know the most about the genetics of big money crops because that's where the research is. So there's a lot of money in researching wheat genetics and corn genetics and soybean genetics. So we knew about this gene. And so we put that into the chestnut tree, and it did indeed detoxify it. But we also developed a strain of blight where we turned off the promoter that causes it to make oxalic acid. So we turned off that gene, and that blight still infected the trees. So it says it's deeper than oxalic acid. Oxalic acid is part of the problem, but it's deeper. I think for an engineered solution, we have got to really understand the molecular biology of the pathogen and tree interface. In terms of trying to make a less aggressive blight, I don't know, we talked about it, but how do you spread it? And that gets back to trying to make that hypervirulence more effective. You know, we could engineer a virus, but I wouldn't want to be the one that released it on the world. We've been there once, I don't want to do that again. Sir?
We have and I know you're familiar with, we have something called a buckeye. It's in the woods that looks a little bit, the burr and the thing, are they cousins or?
No, they're really not related. Other than they're brown and round and shiny. I don't know if carrying a chestnut in my pocket gives good luck or not.
Are there any other questions for Mr. White?
Miller, is there anything the Forest Service is doing in LBL?
There's, we've done a couple test plantings up there. There's a couple of chestnuts we've identified in LBL. I don't know that they're actively doing anything,
They planted,
but they support,
they planted some.
they're cooperative with our work.
Okay.
So I think that's where most of the Forest Service engagement has been, is more cooperating with our foundation in terms of making land and sometimes people available. You know, in eastern Kentucky, we've done work with mine land reclamation. It gives you large pieces of land that is kind of vacant, and it's a great place to plant stuff.
Any other questions? Thank you. I see that Ski is here, and I forgot to ask her if she has any information or news from the Stewart County.
Well, our next meeting will be Tuesday, the day after Memorial Day, and it's going to be a lady portraying Mrs. Henry Worse. Mr. Henry Worse was in charge of Andersville Prison. She's going to talk about her side of the story, the wife. So she's done it before. I think it's going to be a good program. And then we have our joint meeting in June, right?
Right.
Here.
Yes. Yes. Speaking of Stewart County, we will be meeting with them on June the 10th, a Tuesday, at 6 p.m. We will be hosting the Stewart County Historical Society. The program is by Diane Luton Blum and Mike Luton on Dr. O.S. Luton. Bring food for our at the dish table. The meeting will be here at City Hall. Dr. Luton was a physician here for many years. Many of you have had interaction with him, whether he was your physician, whether he was at TVA when you were there, Boy Scouts. You have dated his I don't know.
So that's instrumental in starting.
It's at
our
6 o'clock, though?
celebration.
It's at 6 o'clock at night?
It's at
Yes.
6 o'clock because we're eating food.
It's weekly.
Okay.
He was instrumental in starting the Irish celebration.
So he has roots here. He's from the Stewart County community of Pontiac, I believe. So that's where the Stewart County connection comes with the Houston County connection. And we hope to see you all here at that time. Our list of our meeting calendar, we have July taken care of, August. We have September and October open. We have Mr. Dan Osbrooks for August and Camden Lane for July, the archives in November, and our annual Christmas dinner in December. So if you have anyone that would like to speak at any of these two open positions,
I spoke with Bud Nolan about a week ago, and he wasn't sure, but he would do a program on the history of TVA here in Houston County, in TVA Cumberland.
because we only got two left.
Just not sure if it was September or October. I'll get in touch with him and find out.
All right. Is there a motion to adjourn?
Oh, wait. Anybody that's interested, I'll be at the McKinnon Community Center on Friday at 6 o'clock, talking about the archives and doing a little history of White Oak.
Except it'll be at White Oak, not
White Oak
McKinnon.
Community Center.
Sorry, where is it?
White Oak.
White Oak, I'm sorry, White Oak, yeah, the White Oak Community Center. So if y'all wanna come out, 6 o'clock, if they have an added dish, bring something to eat, good to eat, and I'll be speaking, so.
Okay, is a motion to adjourn?
So moved.
All right.